The next talker is, speaker is Dr. Marko Kostic, who recently got his PhD from UCLA in bioengineering and is working with Warren Grunfest, who's one of the scientific advisors for Pacific Meso Center as well. And we talked about earlier about the issue of using thermal therapy, and I gave some basic information regarding cell cultures, and I alluded to the clinical service that we have in actually very actively doing cryoablations almost on a daily basis in these patients, which has been very effective. And so what Dr. Kostic has been doing is actually working on a method to bring that to intraoperative application. So he's been doing a lot of the engineering work to do that, so he's going to talk about that today.
Thank you, camera. Okay. So as Dr. Cameron mentioned, I'm working basically on a development of design and development of a clinical cryosprayer for a treatment of mesothelioma. And first, I would like to acknowledge the support by Pacific Mesothelioma Center and Pacific Heart, Lung, and Blood Institute for their funding support. Our collaborators, Dr. Cameron, Dr. Claire Cameron, Director of PMC, Dr. Wong, and Dr. Liescu from PMC team, and then my professor, Warren Grunfest, and the undergraduate students, Eric Jong and Gintara Kirzait. So basically today I'll talk about a little bit about why we're doing the research, the development of this system. I'll present some problems in our hypothesis for the research. I'll talk about our novel cryotherapy system development phases. I'll show you some of the simulations that we completed in initial tests, and also our future directions for the system development. So MPM basically claims about 2,000 to 3,000 new cases a year. There are about 2,400 deaths as of 2007. Median survival is about 7 to 10 months if untreated, and treated patients basically 19 months. Recurrence is a problem. It happens in 13 to 62% in local, and then 29 to 56 in distant cases. Time to recurrence is anywhere from 18 months to about 12 years. So current basically treatment is surgery, radiation, and chemotherapy. And surgery, basically the extrapleural pneumonectomy is basically have a survival of median survival of 33.8 months for phase 1 and 2, and median survival of 10 months for phase 3 and 4 cases. Also, there's a PD, and basically surgery is usually part of a multimodal therapy. Radiation is not indicated as a primary therapy, but as an adjunct to radical surgery or palliation of pain. The chemotherapy has very limited effect, what we found in literature, and then we have about phase 1 and 2 responses are 3 to 12% for non-platinum-based agents, and 10 to 20 for platinum-based agents. Phase 3 and 4 cases, 41% of patients have 12.1 months, and then 17% has 9.3 months survival. So the current treatments, based on the literature, basic current treatments have a limited impact on the prognosis of patients with mesothelioma. And the principal problem with surgery is that the primary total excision is impossible, and the identification of remaining cells cannot be achieved with current techniques. So total eradication of disease can rarely be achieved by surgery alone. So we hypothesized that if we develop a system that can eradicate residual disease, that it will significantly improve the impact of surgery on the outcome of surgical treatment of mesothelioma. So basically we asked, what is required for us to accomplish this goal? So based on previous reports in literature, we found that cryotherapy can decrease the recurrence of pleural mesothelioma after surgery, and specifically some studies by Dr. Cameron indicated in a study of 24 patients, they have a high success rate in showing that cryotherapy is actually a good potential treatment for recurrent mesothelioma, and that mesothelioma cells are sensitive to cold. So susceptibility to freezing suggests that cancer cells that remain after resection may be killed using cold gases to freeze the surface of lining of the chest cavity while doing minimal damage to the underlying tissue. Some research suggests that benign lesions require approximately -20 to -30 Celsius to be destroyed, while malignant lesions require much lower temperatures of -35 to -50. So we set our target temperature, treatment temperature, as -35 degrees Celsius. There's a little bit of an overview. Basically, you can see all the anatomical structures that we have to deal with during the treatment. So issues that we have to consider when designing this kind of system is the proximity of vital organs and structures while we are basically doing the treatment. What is the depth of thermal penetration when we do apply the cryogenic material? How large is the surface area that we want to treat? And how effective it is, basically our system will be to deliver the cryotherapy or to reach the target temperature. So with the support from Pacific Meso Foundation, we basically started development of a cryo system that is designed to improve the outcome of surgical treatment of mesothelioma. Our goal was to kill the remaining mesothelioma cells after surgical resection and to basically reduce the recurrence. To achieve this goal, we basically used a cryospray of liquid nitrogen, which is directed at the pleural surface to freeze the tissue to about a depth of 1 millimeter in order to avoid particularly sensitive organs and structures, as we saw in the anatomy slide. So this is our current diagram of a cryo system that will be used for intraoperative use. The pressure will be about 5 psi. Temperature of liquid nitrogen is -196 Celsius. And we're currently using the storage of 0.5 liters of liquid nitrogen. So the center of our system is the liquid nitrogen dispenser. Then we have the dispensing unit, which consists of a hose, the handle that a surgeon will use to actually apply the cryo, and the nozzle, which actually dispenses the liquid nitrogen. On the back end, basically, we have a flow controller that regulates the dispensing of liquid nitrogen, a power supply for the pressure regulator, which regulates the pressure within the vessel, and also power supply for the flow controller. So these are some of the early prototypes of how we envision the system, particularly the handle for the stylus type sprayer, and then also our nozzles, where this will be small and medium nozzle, and we would have a large nozzle. And the difference basically between the three nozzles is the surface pattern that they will deliver to the tissue. So the small, we estimate about 5 millimeter diameter, medium surface pattern of about 20 millimeters, and then a large surface pattern would be over 50 millimeters in diameter. So far, we've developed the design for the initial cryotherapy system. We explored the initial system design using thermal modeling simulations, and some of the results I'll show you in a little bit. We characterized flow rates for 2 prototype nozzles, and then we also conducted the prototype system tests on ex vivo tissue samples. So for thermal modeling, we created basically 3 structures. One was a muscle, bone, and the third one was muscle with bone, but then we also have the fourth scenario where we have muscle and bone, where we actually added the blood flow to simulate the actual patient scenario. So we used the thermal modeling simulation software, and basically what you see here is the propagation of temperature when the basically liquid nitrogen is applied to the surface. We decrease the surface temperature by 5 Celsius each second, and we actually achieved the maximum effective depth in muscle tissue was approximately 3.6 millimeters, and the temperature was at 0 C. Our targeted temperature depth was actually recorded at 1.5 millimeters. So for the bone, you will see it's quite different propagation of a cold. Maximum effective depth was actually 4 millimeters, and that was at 10 degrees Celsius. Our target temperature depth was at 1 millimeter, and we again have the same application of the cryo liquid. Next, we wanted to simulate more of a clinical scenario, so we basically wanted to model the rib cage. Here it says without blood flow, but basically the little circles that you see here, here, here and here actually represent the blood vessels where we'll actually, in the last scenario, we'll actually flow the blood, which will actually be at a body temperature, and you'll see the difference there. So this is without the blood flow. We see that the temperature propagates, but you can see these small blood vessels at the surface. They don't have any effect because the blood is not actually flowing. So the temperature just propagates. And here, we basically reached our target temperature at 0.5 millimeters, basically the minus 35 C, and a maximum effect of that was 3.5 millimeters. Now, when we added the blood flow, which was at 37 degrees Celsius, you can see here how blood vessels basically take away the heat, so they're not allowing as much propagation of the cold in the tissue. So these are just very basic models for thermal propagation that we started with so we can better understand what to expect basically in a depth profile as well as the surface propagation of the temperature when we applied the liquid nitrogen. So we conducted an ex vivo tissue experiment, and we used the porcine rib cage. We used a thermal camera to record the surface temperatures, and we used thermocouples to record temperature depth profiles in the tissue. We used two different nozzle types, high and medium flow rates, and we basically used different exposure times to the cryogenic fluid so we can see how much effect it has on a depth profile. So this is the setup. As you can see, these are the thermocouples. They're actually positioned vertically because we want to get the depth profile. They're needle-based thermocouples. They're approximately 1 millimeter from the surface and then 1 millimeter apart each. So we basically go down to 3 millimeters in depth from the surface of the tissue. Before going to ex vivo, we actually conducted a nozzle pattern test. As you can see here from a thermal camera, we see that a small nozzle pattern of the delivery of the cold versus the medium nozzle, which delivers a lot more liquid nitrogen to the tissue or to the exterior. So we characterize these two nozzle types just so we can better understand flow rates and how it will actually impact the tissue later on. So these are some of the images during our test. We have a picture-in-picture basically with a thermal imaging superimposed on the regular digital camera image. And these are all recorded by the same camera at the same time. So these are, and you can see here's the nozzle, basically the spray of liquid nitrogen coming out right here into vapor. And you can see it right here also. This is the application site right there. So as you can see, we go down to, in this scenario, we went down to minus 47 degrees Celsius for this application. So these are the results that we achieved basically using the small and medium nozzles. We started, for small nozzle, we started at 21 degrees Celsius. And basically went at 5 seconds, we record the surface temperature after spraying, and then we recorded the temperature at a thermocouple 1, 2, and 3, and these are basically 1, 2, and 3 millimeters below the surface of the application site. The flow rates for small nozzle, we measured at 0.397 milliliters per second, and for a medium nozzle, 0.43 basically milliliters per second. On the surface, we universally measured the minus 60 degrees Celsius, as measured with the thermal camera. However, what's important to notice is that the thermocouple at 1 millimeter depth showed very little change after 5 or 10 seconds of application of liquid nitrogen. Basically, only after 20 seconds we went down to 5 Celsius, which is still doesn't provide any effect to the healthy tissue. So these are basically initial results that, we believe that with this system we may be able to avoid the damage to those important structures that are underlying the anatomy of the treated area. So for the medium nozzle, we have similar profile, but we started with a little bit of a higher temperature of the tissue. So here you will see the actual application of in one of the experiments where we actually taken these measurements. So you can see actually the stream of liquid nitrogen coming out. The thermocouples are right there. at a higher temperature, and this is the application site. So this right here, this is the tissue that we used, the porcine rib cage that I showed earlier.
Is this still in the pig, or is this?
No, ex vivo, ex vivo. Ex vivo only, yes. So we're only conducting ex vivo experiments. So we had our development challenges. First one was and one of the most difficult challenges was the hose, actually. Because we were required to have a very low heat loss. The hose actually needed to have sufficient pliability at extremely low temperatures and low weight as to not impede the surgeon's dexterity during the procedure. And we were looking for relatively low cost as well solution. We devised a hybrid solution using off-the-shelf components and some custom-made parts. So we had our development challenges. First one was and one of the most difficult challenges was the hose, actually. Because we were required to have a very low heat loss. The hose actually needed to have sufficient pliability at extremely low temperatures and low weight as to not impede the surgeon's dexterity during the procedure. And we were looking for relatively low cost as well solution. We devised a hybrid solution using off-the-shelf components and some custom-made parts. And now we're able to actually successfully transfer the liquid nitrogen basically through the nozzle from our tank, from the dispenser, without freezing the hose. We used silicone and we used Teflon, which was supposedly rated for a lot lower temperatures, but even Teflon actually froze on us and it became like a solid, like a stainless steel pipe. Initially we thought we may use the stainless steel braided hose, but those are extremely heavy, so we had to have a special support for the surgeon. So that basically didn't. We basically threw that idea out from the table, and then we found a really neat solution. So that's what we're using, and it's been holding up pretty well, actually. The next challenge that we had was actually the flow control, and we're still dealing with this issue. Is basically requires to have a very fine control of liquid nitrogen flow rates, and it needs to have immediate response to surgeon's input, where we basically place the input on the handle, so the surgeon can actually control finally the flow rates based on his assessment of the treated area. So current solution, we have a capability to control flow rates at specific steps. Basically, we have discrete steps. The clinical system design, however, should provide continuous control, or basically smooth control, that is based on thermal camera output. So basically, using the thermal image, the surgeon will be able to know how much more flow rate or how less flow rate needs to have for the cryogenic fluid to the treated area. And we're presently developing this part of the controls. The next and the third challenge that we had was the pressure regulator, because it needed to provide sufficient vessel pressure while maintaining safety of the pressurized dispenser. So we needed to have a feedback control based on internal liquid nitrogen tank pressure. We had to still implement the overpressure safety valve for the rating of the vessel. And it needed to operate at very low or extremely low temperatures. So we designed a custom-made solution using external pressurization system and some of the off-the-shelf safety valves. And we're actually currently in the process of fabricating this solution, and we'll be testing it hopefully soon. But in all these three challenges, the temperature was the key issue, and the humidity of the air causes a lot of components to actually just freeze solid. So these three issues we pretty much overcome, but they still need the testing and a further development. So for our future directions, what we plan to do is system development-wise, we need to expand simulation models with the data based on our measured ex vivo data that we obtained. We need to continue characterizing the liquid nitrogen flow rates with different nozzles and different spray patterns. We only used the 2 nozzles so far that we characterized. We would like to also develop a test system for precise liquid nitrogen flow rate control, which I just mentioned. And we need to design a larger liquid nitrogen storage tank because 0.5 liters may not be sufficient enough for the duration of the surgery or the treatment during the surgery. And then finally, we need to build a clinically suitable system where we wouldn't need a PhD two to run the system actually, or to operate them. For proposed experiments, the clinical trials we would like to test in the chest wall, in animal models, and then humans. We would also like to look at the cellular investigation of the cryotherapy effects, how they actually affect the tissue. And we would like to test the novel fluorescence decay temporal imaging technology, which demonstrated initial quantitative and qualitative differentiation and identification of endogenous fluorophores within the tissue, and it has a good sensitivity and specificity based on a lifetime expected extracted information. So in conclusion, the surgical treatment of mesothelioma has its limitations. Based on a need to improve surgical outcomes, initial therapy system was designed and developed. We conducted some computer thermal models that are used to predict the target temperature depth and tissue thermal profiles. And we conducted preliminary ex vivo tissue experiments showing us some promising results. Further system development is needed for clinical trials. Cellular investigations for cryotherapy effect are needed so we can better understand the physiological processes that basically go behind the application of our cryogenic fluid. Implementation of a novel imaging technique may provide improvements in surgical and cryotherapy treatments. And basically for our animal studies and further system development, we're pending our funding. So thank you very much. I'm a little bit early, but